Hi, I am Luis Herrera-Estrella. I'm the director of the National Laboratory of Genomics for Biodiversity of the Center for Research and Advanced Studies in Mexico. I will be talking about phosphorus, a very important nutrient for plants and agriculture, and this is part of 3 sessions that I'll be presenting for the iBiology program. So phosphorus is an essential nutrient found in many molecules. It is present in phospholipids in the membranes. It is part of RNA and DNA, and it is also part of high-energy molecules such as ADP and ATP. Phosphate is the only chemical form of phosphorus that plant can assimilate from the soil. So in the soil, we have very low levels of phosphate between 1 and 20 micromolar. And inside the plant, we have concentrations that range between 1,000 and 3,000 micromolar. So the plant must take up phosphate from the soil, increasing the concentration about 100 or 1,000 times. So orthophosphate is the only molecule that plants can assimilate. So there are 2 versions of orthophosphate, which are predominant depending on the soil pH. Because of its low solubility and low mobility, Pi is the most limiting nutrient in the soil and most natural ecosystems. Solubility of phosphate depends on pH. At low pHs, acidic soils, phosphorus reacts with iron, aluminum, and magnesium, forming compounds that are not very soluble and cannot be taken up by the plant. At other acidic pHs, it forms complexes with oxides. At alkaline pHs, we find phosphate mainly associated with calcium, and it becomes relatively unavailable to plants. So there is a very narrow range of pHs in which the phosphate is readily available for plant uptake, and it can be assimilated for plant growth. Because of its low mobility and low solubility, roots deplete phosphorus from the soil rapidly, and the phosphate molecules illustrated here as these yellow dots cannot diffuse into the root. So the plant has to search for this nutrient, in contrast to most of the nutrients that freely diffuse to reach the root. In this case, the root has to search for the nutrient, and that makes particularly difficult for the plant to get enough phosphate for its growth. The problem of low phosphate availability is quite important because about 70% of the arable land in the world has problems of low phosphate availability. These red areas in the world show zones of the planet in which low phosphate availability is under the level required for plant production. So low phosphate availability causes reduction of plant growth and disease symptoms like the production of anthocyanins. To prevent this problem of low phosphate availability in agriculture, over 50 billion tons of PI fertilizer are applied annually to increase crop yield. So to cope with phosphate limitations, plant activate mechanisms that facilitate phosphate uptake and scavenging. For instance, when a plant faces a low level of phosphate availability, the expression of hundreds or even 1,000 genes is activated or repressed. The metabolism of the plant is completely reprogrammed, and the plant exudates organic acids and other molecules into the rhizosphere, into the soil that is surrounding the root, to facilitate the solubilization of nutrients, particularly phosphate. And also it undergoes very important developmental changes in its root growth program so that the plant has a different type of root architecture that facilitates nutrient uptake. So you can see here, for instance, how many genes are activated when a plant is exposed to phosphate deprivation. There are hundreds of genes that are repressed, that depending upon the time of exposure to low phosphate increases. So it decreases, for instance, from 1 day of exposure to 10 days of exposure, the number of genes that are repressed, but a larger number of genes are actually activated. So this increase in genes that are activated, it depends on how long the plant has been exposed to low phosphate. These genes that are activated or repressed in the plant allow the plant to alter, to remodel its metabolism in a way in which it can maintain its physiology and biochemistry, but using less phosphate. So many reactions that normally use phosphate, for instance, the conversion of sucrose into different type of carbohydrates, it goes through glucose and then glucose 6-phosphate. When the plant is exposed to low phosphate, it activates pathways which don't require phosphate anymore. The plants also respond by remodeling major components of the plant cell. For instance, the plants degrade phospholipids when they face low phosphate availability and replace these phospholipids in the cell membrane by galactolipids or sulfolipids. These are lipids that don't contain phosphorus, but actually maintain the structure and functionality of the membrane. So these compounds that contain phospholipids like phosphoglycerol, phosphatidylcholine, phosphatidylethanolamine decrease when the plant is exposed to low phosphate, and the lipids that do not contain phosphate, such as sulfolipids and galactolipids, increase in concentration. The degradation, the hydrolysis of these phospholipids requires the participation of phospholipases. These are enzymes that break down phospholipids, and one of these enzymes is phospholipase D zeta 2. This gene was identified in our lab, and as you can see here, the expression of this gene, this is a fusion between the regulatory sequences of the phospholipase gene and the gas reporter gene, which produces a blue staining. So when the plant is under phosphate, normal phosphate nutrition, the plant doesn't express the gene, or it expresses it at low levels. But when the plant is exposed to low phosphate, then the gene is activated in the leaves and very strongly in the roots, so that the phospholipids are hydrolyzed and phosphate is released for more important, more essential biochemical processes than just maintaining membrane structure. When you mutate this gene, when you have a plant with sufficient phosphorus nutrition and you expose it to low nutrition of phosphorus, the level of phosphatidylcholine decreases because this phospholipid is degraded. When you mutate the phospholipase D2, this decrease is much less, suggesting that this enzyme is directly involved in the process of phospholipid degradation activated by low phosphate availability. So how this gene is activated? So we did some studies to analyze which are the regulatory components in the transcriptional regulatory sequences, in the promoter sequences of this gene, to see which elements were required to activate this gene. So this is a work done in our lab and several other labs that have identified elements that are required to have an activation. So this is the expression when the plant is grown in high phosphate, and when we put it in low phosphate, the expression of the gene increases drastically. We can remove parts of the regulatory sequences gradually until we find a sequence that when it's removed, the gene is inactivated. And we found that there is an enhancer element. Enhancer elements are transcriptional elements in the DNA that activate the expression of genes. And this enhancer element is constituted by specific signals that are conserved between many plant species. And this contains a site that is called PHR1 binding site. And I will explain what PHR1 is in a few moments. So these elements are required for the activation of genes that are responsive to low phosphate. And this program has to have some components that are important for the transcriptional activation. So as I mentioned earlier, hundreds or thousands of genes are activated when the plant is exposed to low phosphate. So PHR1 is a transcription factor that plays a very important and central role in the activation of these genes. This transcription factor is a protein that binds to the sequences that I showed you before that are required for the activation of these genes in response to low phosphate. PHR1 activates the expression of about 200 or 300 genes directly, and these genes indirectly activate thousands of genes that are required for the adaptation of the plant to low phosphate conditions. So how this system works, how is phosphate detected? So one very interesting recent advance in the understanding of this mechanism is the study of two proteins, PHR1, which is the transcription factor that I just mentioned in the previous slide, and SPX1, which is a repressor of the transcriptional activity of PHR1. So when the plant is exposed to high phosphate, PHR1 and SPX1 form a complex which is not active. So the genes that are responsive to low phosphate are not activated and they have low transcriptional activity. When phosphate is low, the interaction between PHR1 and SPX1 is disrupted, and then PHR1 can bind to these sequences that are specific for the activation of low phosphate responsive genes, and then these genes are activated. So this complex of SPX1 and PHR1, this interaction is dependent upon the concentration of phosphate in the cell. So low phosphate, no complex, transcriptional activation. High phosphate, the complex is formed, and then there is no activation. This system was reported by the laboratory of Javier Pasares and Ping Wu in Spain and China, and it makes a significant progress in our understanding how these mechanisms are activated. Now, plant responds to low phosphate in two different manners. One is a systemic response, which depends upon the concentration of phosphate inside the plant. And that signal remodels the expression of genes in every cell of the plant. And there is a local response in which only the cells that are in contact with low phosphate in the external media respond to the signal. So there are two different responsive mechanisms. The systemic response is mainly regulated by PHR1, that is the central regulator of transcriptional activation to low phosphate. And the local response is still sort of mysterious because we don't know exactly how phosphate is sensed in a local manner and how this activates very important developmental processes. So split root experiments allow the identification of genes that are regulated by local and systemic signals. So in these experiments, what you do is you have a plant and you split the root systems into two. In one, half of the root, you put it in a media containing high phosphate, and the other half, you put it in media containing low phosphate. If the signal is systemic that regulates that gene, the presence of high phosphate in one side will inactivate the expression in the half of the root that is exposed to low phosphate. So that's very important. If the signal is local, in spite of the plant having high levels of phosphate, this root will respond to the low phosphate signal in a local manner. And this is an example of what happens if you have the plant exposed to high phosphate, the expression of this gene is low. If you expose to low phosphate, the expression is high. If you do the split experiment and this gene is regulated by a systemic signal, in both the half of the root with phosphate is low, and in the half of the root without phosphate is low. If the signal is local, you have low expression when the whole root is exposed to high phosphate, high expression when the root is exposed to low phosphate. But when you do the split experiment, in the half of the root with phosphate, expression is low, but expression in the half of the root that has low phosphate is high. So this suggests that indeed there are two different molecular mechanisms regulating expression in the plant when it responds to low phosphate. Now, how the plant maintains the homeostasis of phosphate. So we saw in the previous presentation that you can add a nutrient in amounts that are limiting, then you reach a maximum, which allows the plant to achieve maximum productivity. But if you overfeed the plant with the nutrient, then it's toxic. And this is particularly for phosphate. If there are excessive amounts of phosphate in the plant doesn't like it, it doesn't grow. So there have been a number of mutants that have been isolated, like PHO1 and PHO2, which affect the responses of the plant to phosphate homeostasis. These mutants were isolated by Manny Delhaize and Peter Ryan. And these mutants, one has, this is the wild type, the content in the shoot and the root of phosphate. So they have more or less similar levels of phosphate. The PHO2 mutant overaccumulates phosphate in the shoot, and the PHO1 mutant doesn't accumulate phosphate in the shoot. It has reduced amounts of phosphate in the shoot, although it has normal amounts or even increased amounts of phosphate in the root. If you cross these two mutants, PHO1, the PHO1 phenotype, meaning low phosphate in the shoot and high phosphate in the root, is maintained, suggesting that these two genes act in a common signaling pathway. So, and both having high phosphate or low phosphate in the shoot are not good for the plant. If you have too low, the plant doesn't grow. If you have too much, the plant doesn't grow. So we need to have an homeostasis on how much phosphate is in the plant. And there have been very interesting advances by several groups in the world studying how these proteins act. It's been discovered that PHO2 is a protein that participates in the degradation of the proteins that allow phosphate transport. So this protein, when it's activated, is an ubiquitin activase that sends these transporters for degradation. So when the plant is in normal conditions, PHO2 destroys a certain amount of transporters, both the transporters that allow the entry of phosphate into the epidermal cells, and also the transporter that allows the transport of phosphate into the xylem to move up into the plant. So PHO1 is the transporter that allows the entry of phosphate into the xylem. When PHO1 is mutated, roots have normal amounts of phosphate because the other transporters are allowing the entry of phosphate into the root. But since we don't have the transporter in charge of transporting phosphate into the xylem, then there is no transport to the shoot, so the shoot has low phosphate. When we have a mutation in PHO2, which I said before participates in the degradation in maintaining a reduced level of transporters in the plant, there is no degradation of transporters that allow the entry into the root, and there is no degradation of transporters that allow the entry of phosphate into the xylem. So then we have a very large amount of phosphate accumulated in the shoot. So this is a cycle which is very interesting and has been studied by different groups and have been made very interesting progress. Another interesting part of this story is that the accumulation of PHO2 is regulated by a microRNA. MicroRNA are molecules that are encoded by genes that actually don't produce proteins, but either produce small RNAs that regulate the expression of genes by either triggering the degradation of the messenger RNA or by preventing the translation of the messenger RNA. So what happens is that when you have high phosphate, there is low levels of the microRNA 399, so the plant has enough quantities of PHO2, and PHO2 prevents the accumulation of the transporters, so the plant maintains a phosphate homeostasis. When we have a low phosphate, the transcription of microRNA 3939 is increased. The microRNA is transported via the phloem to the root, and in the root, we inactivate the expression of PHO2 by decreasing the level of the messenger and the translation of the messenger. So we accumulate the transporters, and the plant takes up more phosphate, enhancing the capacity of assimilating phosphate. So now, the deficiency of phosphate also alters root architecture. This is a very dramatic phenomenon that occurs in most plants in different flavors. Some plants respond in one way, and other plants respond in another way. And this is what happens in Arabidopsis. When you have the growth of Arabidopsis under high phosphate, the plant produces a long root which has branches in a gradual formation of lateral roots. And there are very few lateral roots near the root tip. When you expose to low phosphate, you have a shorter root which is very highly branched. It produces many lateral roots. And not only produces lateral roots, but it produces, this is the normal root hairs, the cells which actually are the most active in nutrient uptake in the root. When you expose to low phosphate, you increase the density and the length of the root hairs. So this process is triggered by low phosphate. Is a very dramatic change in the developmental pattern of roots in Arabidopsis and many other plants. So why is this change? How do we explain why these plants react in this way? Well, the possible explanation is that phosphate, because it has a low mobility, it accumulates in the upper layers of the soil. So in the first 20 centimeters, we have high concentrations of phosphate. If we go 20 centimeters below, we have less phosphate. And if we go 40 centimeters below the ground, then we have much less phosphate. So because this accumulation of phosphate in the upper layers of the soil, it will be more favorable for a plant for phosphate uptake to have a shallow, highly branched root system than a long root system that will be more efficient in taking up water than nutrients. And this is particularly important for phosphate because nutrients that freely diffuse in the soil have similar concentrations independently upon the depth of the soil. So how this program, this change is activated? When in Arabidopsis, my group has done so much studies showing that the developmental pattern of growth of the root changes from an indeterminate pattern in which cells keep dividing infinitely to a pattern in which it arrests growth and cells differentiate to become highly specialized in nutrient uptake. So cell division, cell proliferation, which is shown here as the blue dots, these cells are undergoing cell division, is arrested in roots that are exposed to low phosphate. And the stem cells that organize the growth of the root and give rise to all the different cell types that form the root disappears in a root that is exposed to low phosphate. And these low phosphate roots become specialized in the production of high affinity phosphate transporters. This is the root tip of a plant exposed to low phosphate. So you can see that there is a very high level of expression of high affinity phosphate transporters that help the plant to get phosphorus from the soil, and also in the expression of phosphatases that help the plant to get orthophosphate from organic molecules. These genes are not expressed in plants exposed to high phosphate. These developmental changes can be quite dramatic. For instance, these are protoid root formations. They are clusters of roots that are formed in the roots of certain type of plants, such as lupinus, in which you have a very dense formation of lateral roots that are specialized in the uptake of phosphate. They accumulate high levels of phosphate transporters, they excrete a lot of organic acids, they produce a lot of phosphatases. So these changes are very interesting, but it can be many different ways in which you can make a shallow system. For instance, in the case of beans, the developmental change is that the root, instead of growing down, it grows vertically. So it doesn't change the number of lateral roots, but rather it changes the spatial distribution of roots. In maize, you have more formation of crown roots. And as I mentioned before, in lupinus, there is the formation of cluster roots. There are demonstrations that the group of Jonathan Lynch has studied what is the influence of root architecture in bean plants. So he has found that there are bean genotypes that have shallow roots, others that have intermediate root architectures, and others that have deep roots. So what happens when you expose these different genotypes of plants to different concentrations of phosphate? If you have an uniform concentration of phosphate along the plane of the soil, there is almost no difference in plant uptake. But if you have a gradient, as it happens in most of the soils, if you have low phosphate availability and you have phosphate availability only on the top layers, but not on the bottom layers, this plant becomes more efficient in phosphate uptake, and this one that has long roots is less efficient. So this is a clear demonstration that root architecture has a very important role. And perhaps the most definite demonstration of the importance of root architecture in phosphate uptake is the study of the phosphate uptake 1 QTL. QTLs are quantitative traits that contribute to a certain characteristic of a plant, and that can be mapped through crosses in different plants. So in rice, people has been studied this particular QTL, which was found in wild species of rice, such as Oryza sativa casalat and Oryza glaberrima. And these plants contain this QTL. When this QTL was mapped, it was found that it was present in wild rice species that are efficient in phosphate uptake, and they are naturally adapted to low phosphate soils, but they are absent in modern cultivars. So these plants during breeding have lost the capacity of facing low phosphate. And the demonstration that this is the case is that if you express these genes from the wild rices into cultivated rices, you alter root architecture, making a highly branched root architecture, and you produce more grain under low phosphate than the original genotype. So how lateral root formation occurs? Lateral root formation is activated by auxins. It's a hormone that has an activity of a morphogen. It triggers the development of many organs in a plant, and changes in the gradients of auxins triggers the formation, for instance, of lateral roots. Lateral roots form in the primary root by the cell divisions of cells that are normally sleeping, that they are dormant, and then they activate the formation of lateral roots. So what happens when you expose a plant to low phosphate? Does the concentration of auxin increases, or the sensitivity of the root increases to auxins, and that's why it forms more root? We did some experiments to study what were the changes in auxin, free auxin concentration, and we found that the concentration of auxin is very similar in plants grown on high phosphate or low phosphate. However, when we expose to auxins, low phosphate and high phosphate roots, the low phosphate roots react to auxin concentrations that are very low. For instance, 10 to the 10 molar activates more lateral root formation in a low phosphate exposed plant. Under high phosphate plants, you need about 1,000 times higher concentration to trigger the formation of lateral root. This suggests that plants exposed to low phosphate are more susceptible to auxin action. And why they are more susceptible? We found that low phosphate activates the expression of TIR1. TIR1 is the receptor, one of the receptors for auxins, and when you have more receptor, the cells become more susceptible to the presence of auxins, and then it triggers the expression of the gene, particularly in the pericycle cells where lateral root formation takes place. So this activation of TIR1 makes the cells more susceptible to auxins and then form more lateral roots. How this becomes true? Well, there are inhibitors of root action. There are transcription factors that inhibit the activity of auxins, and these inhibitors, these repressors, are destroyed by the activity of TIR1. When you activate TIR1, these repressors are destroyed by the ubiquitin pathway, then the cells can respond to auxins. If low phosphate plants are more susceptible to auxins because TIR1 is activated, these repressors should be more rapidly destroyed. In this case, the repressor is linked to the Gauss reporter gene, so that the blue staining represents the amount of this repressor. If you have high phosphate, the level of the repressor is high, and if you expose to different times, the repressor is still there. If you have low phosphate, already at the beginning of the experiment, the inhibitor is in lower concentrations, but it is very rapidly degraded, suggesting that indeed the activation of TIR1 leads to the destruction of these repressors, and then the plant can respond to auxins. So we develop a model in which this TIR1 modulates the activity of the transcription factors that activate auxin responses. So we have the activator and the repressor bound together in a complex in the DNA, and this is inactive. When we activate, we increase the expression of TIR1. TIR1 triggers this repressor into degradation, and then we liberate the transcription factor that activates transcription so the cell can respond to auxins, and this brings as a result the production of more lateral roots. So now, is phosphate only a metabolite, or it also has a role as a signaling molecule? So this has been a huge debate whether all the effects we see is because the plant doesn't have enough phosphate and it reduces growth because it doesn't have enough phosphate, or it is actually a signal that can trigger responses independently of its action as a metabolite. So if phosphate is a signaling molecule, one should be able, this is a representation, a cartoon of how a plant grows, an Arabidopsis plant grows on low phosphate. So if phosphate acts as a signal, we should be able to obtain mutants that do not respond in this change of developmental program, even if it's under low phosphate conditions. If it's only a metabolite, we should not be able to find these mutants. And what happens is that we can find these mutants. This one is having a very long root compared to the others that have short roots. And this is how it behaves in the presence of low phosphate. So this strongly suggests that phosphate is a signal molecule. Moreover, there is another molecule which is similar to phosphate, which is phosphite. It's a non-metabolizable compound, but it also triggers these changes in architecture. So even if it's not metabolized, it acts as a signal, suggesting that indeed phosphate is not only a metabolite, but it's also a signaling molecule, which is similar to what happens to nitrate and ammonia, which are also as a signaling molecules. Now, this change in root architecture is regulated by systemic or local signals. So in this experiment, we have a Petri dish which is divided in two halves. And on top, we have high phosphate, and at the bottom, we have high phosphate. So this plant is exposed to high phosphate, and it produces a long root. But what happens when we feed with high phosphate the shoot? So the plant has a high phosphate concentration, but then we expose the root to low phosphate. What happens is that the change in root architecture is triggered, suggesting that these changes in root architecture are regulated by local signals and not systemic signals. And these mutants probably will help us to identify the signaling components for this local sensing mechanism. And finally, phosphate is not only altering metabolic pathways, the biochemistry, and the cell biology of the plant, but it probably also has important changes in the epigenome. The epigenome is the changes that allow the genome to alter its function without having mutations. This is through methylation of DNA, through changes in the structure of the chromatin that prevent or activate the expression of genes depending upon the life of the organism. So this suggests that some organisms can be primed or prepared to better respond to an environmental change once they have previously been exposed to that environmental change. So we have been studying what happens when the plant is exposed to low phosphate, and we have found that there are specific changes in the methylation pattern of the plant, and that mutants affected in methylation are hypersensitive to low phosphate, suggesting that methylation also plays a very important role in the response to low phosphate, and probably we could use these epigenetics to prepare seeds to better face low phosphate conditions in agriculture. So finally, just a few words to sum up what I have been saying. The plant response to low phosphate is quite complex. A better understanding of this response will help us to develop more efficient plants in uptake and utilization of phosphate. And it is a very complex. We have talked about the interaction of roots with microorganisms, the secretion of organic compounds, the expression of phosphate transporters, systemic and local signaling, regulators that activate different biochemical and morphogenetic programs. And as in the case of rice, genetic diversity in many crops will serve as the source to develop germ plants better adapted to low soils. So there are QTLs that have been identified for lateral root length, lateral root number, root hair density, the type of root architecture in maize and beans and rice, sorghum, and so on. So understanding all these mechanisms will help us to finally design plants which are better for agriculture and will allow us to do more sustainable agriculture. And just finally want to thank my group of research that have contributed to many of the topics that I talked to you today. And I want to thank the iBiology program for this opportunity to talk to you about phosphorus, a very important element for plant nutrition and agricultural production. Thank you.